All right, guys. So last time, in one of the last math review sessions, we started talking a little bit about what was called primary productivity. And I told you that whenever you hear the word producer, production, productivity, what should you think about? Photosynthesis. What's getting produced during photosynthesis? Sort of. What are the big two products of photosynthesis, though? Glucose and oxygen gas. Okay? Glucose and oxygen gas. And what's getting fixed or used up? Carbon dioxide. Okay? So I put this equation on the board last time around. And I made an analogy to like a paycheck. You're getting paid $5 an hour and you work for 40 hours. $5 an hour for 40 hours, this would be $200, right? What do we call that $200?
Illegal. That's below minimum wage.
That's okay. It's gross pay. Okay? Gross pay. But then, Uncle Sam is going to go Robin Hood on you and steal all your money. And you're going to end up, instead of 200, maybe you end up with 130 or something, right? We call that net pay. So up here in this formula, NPP is net primary productivity. That's equal to gross primary productivity minus, in my analogy here, who's the tax man? Who's using up what photosynthesis is making? Respiration. It's using the glucose, it's using the oxygen, right? It's using both. So we want to think of the respiration like a tax man. So here's our problem here, 38. Water with algae inside is placed into a completely dark environment. If the initial dissolved oxygen concentration of the water is 12 milligrams per liter, and the dissolved oxygen concentration after an hour is 8 milligrams per liter, how much oxygen is consumed by the plant per hour? Well, if it's in the dark, the plant is only doing what process? It's our respiration. It can't do photosynthesis. There's no life. So all we're going to do here is subtract, right? The oxygen levels go down by how much? 4. And that's all we do. We subtract 4 milligrams per liter. That's how much respiration the plant does, correct? Per hour. 4 milligrams of oxygen used per hour, per liter. Then 39, a bottle of water with algae inside is placed into a sunny environment. Starts off with 12 milligrams per liter of oxygen, and after 1 hour in the light, it's got 25 milligrams per liter. How much net oxygen is produced? Now this plant that's in the light, what's happening with that plant? What's it doing? Still doing some respiration, right? But it's also doing photosynthesis. It's doing both. Which one's it doing faster? It's doing photosynthesis way faster because we know what's happening. How do we know that? From the numbers. Oxygen levels going up, right? If respiration was happening faster, oxygen levels would be going down. So to get the net amount of oxygen produced, we're going to go 25 minus 12 and get 13. That's how much oxygen, net oxygen production per hour. Now, something we didn't address last time is this second part of this question. How much net carbon is fixed? If oxygen is being released, carbon is being taken in, right? If you look at your formula sheet, get you one of those in front of you. There are two formulas on this formula sheet that you need to know how to use. And one of them looks like this. That's one of them. And the other one looks like this.
What these ones are going to give us?
This formula sheet you can use on the whole test. It's this exact formula sheet. This is not a formula you need to memorize. You just need to know what it's for. All right, so here's the deal. we know how much oxygen is released. So to get the amount of carbon fixed, these two equations will let us convert from oxygen released to carbon fixed. And the way they're going to do that is, notice that we just calculated how much oxygen was released, 13 milligrams per liter, the net oxygen released. Notice this first formula says take your amount of oxygen in milligrams per liter, multiply it by 0.698. And what that does is that converts milligrams to what? Milliliters of oxygen. Normally when we're dealing with gases, we want to deal with like volume units instead of mass units, right? We like to measure gases like we measure liquids in milliliters. So all this does is convert milligrams of oxygen to milliliters of oxygen. The second formula takes the milliliters of oxygen, multiplies it by 0.536, and that converts between oxygen and carbon. Because, you know, in the equation for photosynthesis, we have coefficients. It's kind of a stoichiometry problem, right? We know for every one mole of oxygen that we give off, how many moles of carbon got used up. Think about the equation for photosynthesis. For every one oxygen that's given off, how many moles of carbon dioxide are taken in? What's the equation for photosynthesis, guys? 6 CO2 plus what? The equation for photosynthesis. 6 CO2 plus 6 H2O yields glucose, 1 glucose, plus 6 O2. So there's a 6 in front of the oxygen, and there's a 6 in front of carbon dioxide. So it's a 1-to-1 molar ratio. This takes all that into account. You don't really know that. You don't really need to know that. You take your milliliters of oxygen per liter, multiply it by this, and that converts it to how much carbon was used up. So by knowing how much oxygen was given off, you can calculate how much carbon was taken in. That's essentially all we're doing. We know how much oxygen was given off. So let's take this 13 milligrams per liter of oxygen. Use this first equation first. So we're going to take our milligrams of oxygen per liter, multiply by 0.698. Calculate that out. That gives us 9.074. And that's milliliters of oxygen per liter. Well, then we're going to take this answer and we're going to substitute it right there into that second formula. And that second formula says take your milliliters of oxygen times 0.536 and that gives you how many milligrams of carbon were taken in, were fixed by the plants. And that comes up to be 4.86. One thing to be real careful on, the test is going to tell you what place to round your answer to. It's going to say round your answer to the nearest hundredth or tenth or thousandths. The math questions on this test are not multiple choice, they're bubble in. So if it says round to the nearest hundredth and you round to the nearest tenth and you bubble it in to the tenth, it's going to kind of wrong. So pay attention to the questions. Make sure you pay attention to what place it says to round to. All these equations do, though, is simply allow you to take how much carbon was, or how much oxygen was given off, and using these two equations, use the amount of oxygen that was released by photosynthesis to figure out how much carbon was taken in by photosynthesis. That's the purpose of those two equations.
Do you have to bubble in the units?
No, just the math time. If they put a unit thing on there, that would probably would be multiple choice. Typically not, but typically they just want the number. One other thing on this part, and then we'll get to the topic that I really wanted to get to today, which is cells. All right, the last part of this equation says, the last part of this question, assume the bottles described in 37 and 38 are the same bottles. That should say 38 and 39, I think. This bottle was in the dark. The plant used 4 milligrams of oxygen per liter per hour. This one was in the light. It released 13 milligrams of oxygen per liter in an hour. And I want to know what's the gross productivity. Got to go to this equation. So let's solve that for GPP. Yep, so GPP equals NPP plus R, right? And just do a little algebra, solve that out. That should be R. So how much total oxygen did this thing make? This was the net, 13. That was the respiration, 4. So the plant, what this is telling us is that the plant made 17 milligrams per liter of oxygen, but it used up what? It used up 4, so it ended up 13 to the good, right? That's the net. And that's all that formula means. Remember, when you see production, producer, productivity, think photosynthesis, and think the plants are producing sugar, oxygen gas. And then those two formulas, right there on the top, kind of middle of your formula sheet, that's what those are about. It's calculating how much, knowing how much oxygen was given off, calculating how much carbon was taken in. All those two formulas do. I want to talk mostly today, though, about cells. That was one math thing, though, that I wanted to wind up with first. So with cells, first of all, remember that the most simple cells on Earth are prokaryotic ones, thought to be the first cells on Earth. And the only things alive on Earth today that have prokaryotic cells are bacteria. And we know literally what does prokaryotic mean. For the nucleus. Oh, it's right up there on the thing. We know they lack a membrane-bound nucleus, but they do have DNA. And what does their DNA look like? Single circular chromosome, which it says on the next one. And in addition, what small pieces of DNA do they sometimes have to go with that? Plasmids, which they will use from time to time to do what? Conjugate and then swap, right? And we said that's sort of like bacterial what? Sex, because what's the purpose? To create genetic variation, diversity. Remember that prokaryotic cells don't have the membrane-bound organelles. No mitochondria, no chloroplast. Pretty much everything they do has to happen where? In the cytoplasm.
So, question. Okay, so a lot of the antibiotics we use today are not working on bacteria. So, can we just can we just create new antibiotics, or is there going to come a day where bacteria kill us all?
Maybe. I mean, there's several things like tuberculosis, like some forms of staph, that are resistant to almost all antibiotics we have. Now, hopefully, we'll be able to make some to counteract that. But in Russia, they're actually using bacteriophages to treat bacterial infections. So they're infecting you with a virus to kill off the bacteria that you're infected with. Well, no, because bacteriophages don't infect our cells, they just infect bacterial cells.
So you're just talking about virus reference?
No, it'll go away because it doesn't infect your cells, it just infects the bacterial cells that are in you.
So like once those die, they just
Then they go away. So that's one way we might go eventually, but we'll see. I think we'll figure it out. Also remember that prokaryotes have ribosomes. They're not membrane-bound organelles, right? They have to have ribosomes because what wouldn't be any good without them? DNA wouldn't be any good, right? Because they make the proteins. Got to have those. Their structure is different than a eukaryotic one, though, slightly. Their membrane is made pretty much the same as your membrane, phospholipid bilayer. And bacteria do have a cell wall, which is mostly there to keep them from what? What's the wall mostly there for? Not so much about drying out. It's to keep them from bursting when they're where? In fresh water. Because what would happen if they were in fresh water? Water would go in, and what would build up? Turgor pressure, and they would pop if there was no wall there to counteract that. So eukaryotic cells are everything on Earth except bacteria, all cells at least. It means true nucleus. We know that our cells, as eukaryotes, have an enclosed nucleus, enclosed by a phospholipid membrane. Our chromosomes, there's usually several of them. And that's how many?
46.
46, and there's many, they're not circular. We know that we have lots of membrane-bound organelles, like mitochondria, Golgi body, the ER, the rough endoplasmic reticulum, lysosomes, and so on. And we know that some advantages of being eukaryote, or eukaryotic, is that each one of those little organelles takes on a certain way. Function. And it becomes very specialized at that function. And it divides at the labor. So if you specialize and you divide at the labor, you become more efficient. So our cells are way more efficient than a bacterial cell. It's like, you can hire people that call themselves jacks of all trades. And if you have a lot of work to do at your house, they might fix your plumbing, they might do some carpentry work, they might do some wiring. They can do all of them, but are they probably masters at any of them? No. Probably not. That's kind of like a bacteria. But your eukaryotic cell has a built-in plumber and a built-in mechanic and a built-in electrician, and they know how to do things right and well. They're specialized. Also remember that eukaryotic cells tend to be way bigger or way smaller than prokaryotic ones.
Bigger.
Way bigger. Mm-hmm.
So if these bacteriophages that they use in Russia, what happens if they mutate to be able to infect human cells?
Good question. No. All right, another thing to think about is that cells are small. And the reason they're small is because as they grow, what increases really fast?
The surface area to volume ratio.
As they grow, their volume increases way faster than their what? Surface area. And think about a cell. What part of the cell is like its surface area?
The membrane.
It's membrane. And the volume would be like the inside part. Remember a long time back when we did the little pink cell lab? We had the little cells with phenolphthalein in them, and they were pink, and we soaked them in what? We soaked them in an acid. And when phenolphthalein comes into contact with an acid, what happens to it? It turns clear. Okay? So they were pink to begin with. We put them in an acid, they turned clear. And what we saw in the big cells is what happened.
They were still pink in the middle.
There was a lot of pink. There was a lot of places where the acid never got to. But in the little cells, what happened? They were clear all the way through. Because small cells have a higher surface area to volume ratio, which means they're better at what? Transporting things. Bigger cells have a lower surface area to volume ratio, and they're not as good at transporting things. And that's why cells don't get big. For a cell to be efficient, it needs a high ratio of surface area to volume. So it has to stay small to keep that. That's important. All right, let's talk about the membrane here for a minute. We know the cell membrane is made of these guys, phospholipids, in what arrangement? A bilayer. And we know that these phospholipids are important because they have what property?
They are
Put one word on it. Amphipathic. And that means they have one side, this side, the fatty acid tail side, that's mostly what? Non-polar. And they have another side, this side, where the phosphate group is, that's polar. So that when we put these things in water, they arrange themselves like this, with the polar head part being pointing toward the water, because the cell's filled with water inside and outside, and the tail part, the nonpolar part, on the inside of the layer pointing toward the other nonpolar part. And that's our phospholipid bilayer. And in every known cell, we've got the same basic arrangement. Another piece of evidence that scientists would used to say would all come from what? Common ancestors. Doesn't matter if you're bacteria, or protist, or fungus, or plant, or atom. You all have a common ancestor. All right, and the model that we use to explain our cell membrane, we call it the fluid mosaic model. It's fluid because the phospholipids are kind of liquidy, right? And it's a mosaic because what's jumbled in there? Proteins. Now, it looks like it's jumbled in there. Is it really jumbled in? It's arranged, right? But lots of different proteins. We have these things right here that I like to call channel proteins. What process do channel proteins carry out? Facilitated diffusion. And what kinds of things do they move in and out of the cell? Charged and also maybe large things. Facilitated diffusion, is it a passive or an active thing? Passive. Because it's moving those things which way? Higher concentration, lower concentration. There are similar proteins, which, we'll just say this one for right now, that are called carrier proteins. What do they do? What process? Active transport. They're pumping things which way? Against the gradient, from low to high. What's the most famous one of those in your body? The sodium potassium pump, which is really important where? The nerves. Helping you to generate gradients in your nerves. We might also talk about these guys, which I'm going to call receptor proteins. And we know they're receivers for what? Ligands. Might be hormones, pheromones, neurotransmitters, all kinds of cells signal receivers. And then finally we got these things sticking up here at the top, which are part protein and part sugar. Might be called glycoproteins. We've been calling them MHC. Sometimes we call them name tags. And we said that those things, number one, they identify your cell to what part of your body? Your immune system, okay, your white blood cells. They also might let one cell connect and communicate with another cell, like the MHC class 1, for example. What kind of cells have those? MHC class 1s. All cells, and their job is to display what? Antigens from inside to what? To either helper T cells or killer T cells, and say, hey, I'm okay, or hey, I'm sick. You need to do what? You need to kill me before I make everybody else sick. So that's the purpose of MHCs. What are these yellow things spread out in the cell membrane?
Protein.
Those are molecules of cholesterol. Cholesterol's job in your cell membrane is to make it a little what? Thicker, a little bit less liquid, give it a little structure. And we know that cholesterol falls into what category of organic compounds? What kind of lipid? It's a steroid. It's what all the steroid hormones are actually built from. Testosterone and estrogen start out as cholesterol. And they get some add-ons or some takeaways to become one of those two. So what kinds of things can go through the cell membrane, no problems? Small and uncharged, non-polar, right? Things like steroids.
So uncharged particles can even pass in the polar parts.
Yeah, they can go through without much problems. The things that have a harder time getting in and out are the ions and the big things, like proteins, for example. And there has to be another way to get them in, either through a channel or through a carrier to get them in and out. All right, we talked about the nucleus of light. We know that the DNA is enclosed by this nuclear envelope or nuclear membrane, and it's actually a double phospholipid bilayer. So it's actually 4 layers of lipids, double phospholipid bilayer. It's got pores in it. It's usually attached up to the ER. Why does that make sense? That the nucleus is attached to the ER. Yeah, because the nucleus has to send out what molecule? RNA. And the RNA, most of it goes to where? The ribosomes that are on the rough ER. So it's the placement thing makes sense. We know that the chromosomes are in the nucleus. And again, how many chromosomes in a normal human cell? 46. And how many of those are autosomes?
44. Yeah.
44, and the other 2 are sex chromosomes.
So the rough ER is where transition happens.
Yes. All right, the next thing I want to talk about is this guy. What did we call this whole diagram? Yeah, but more than that. We call it the endomembrane system. Endomembrane system. And I want you to think about what the endomembrane system is. The endomembrane system typically starts at a ribosome. A lot of your ribosomes are sitting on top of what organelle? The rough ER. They make the proteins, which we can see being made like right in here, this little chain of spheres. What's each one of those spheres? It's an amino acid, right? And then once it's been made, it drops into the rough ER, which is sort of like a passageway in the cell, right? That's its job to transport. Once the protein goes in, what happens to it? It gets bent and shaped into the right structure. It might get some stuff added to it, like maybe some sugars or some lipids or something. So it becomes functional. It moves through the cell and eventually gets to the end of the rough ER where what's happening here? Invagination. And eventually it gets a lipid bilayer around it. And we call that a vesicle, a secretory or transport vesicle. Where's that going to go? Before that. It's going to go to the Golgi complex. It's a Golgi body, Golgi apparatus. And I've compared the Golgi to what kind of factory-related thing? It's like a warehouse. It goes there, sits there for a while. While it's sitting in the warehouse, it may get tinkered with a little bit. It may get some more things added to it or taken away from it to get it ready. Once it's ready, this little vesicle will again break off the Golgi. By the way, where's the Golgi always going to be at in the cell? Near the membrane, right? Somewhere near the membrane. The little vesicle breaks off again, eventually bumps into the membrane. What happens when a phospholipid vesicle bumps into a phospholipid membrane?
Phospholipids fuse into the membrane.
Yeah, they fuse. The membrane grows a little bit. What happens to the thing that was inside, like the protein? It gets pushed out. And we call that what? Exocytosis or secretion. That's the way things get pushed out of your cells. So the endomembrane system starts at the ribosome and then goes to where? What's part 2? Rough ER, to a vesicle, to a Golgi, to a vesicle, to out. That's essentially the pathway of the endomembrane system. Now remember, you also have some ribosomes that are where? Just floating around in the cytoplasm, not attached to anything. What do?
They make things for themselves.
They make proteins that cell is going to use, not that it's going to send out. The things that it's going to send out are made by ribosomes on the rough ER. The ones it's going to keep and use for itself, made by those non-attached ribosomes. Free-floating. Not be a better way of putting that. So there's our endomembrane system. A pathway. It's about secretion. Sending things out of the cell. Let's talk about lysosomes here for a couple minutes. Lysosomes are essentially bags of what? Enzymes. Hydrolytic enzymes. What do we know hydrolysis means? To use water to break things down, right? Hydrolysis. And one of the most important things that the lysosomes do is after a cell phagocytizes something, takes it in, say a white blood cell phagocytizes a bacteria, it's going to put it in a vesicle, and then what's going to happen? The lysosomes are going to come over to that vesicle and dump what into that vesicle? Enzymes. And what we call the process that's going to happen next? Intracellular digestion. It's going to break it down, kill it. But lysosomes also help you to get rid of other things, like old cells of your own that need to be gotten rid of. Certain germs, certain viruses. They even play a role in things like metamorphosis. When the tadpole's tail needs to go away, it needs to make legs. Essentially, the lysosomes break down the tail, and all the pieces get used to remake what? Legs. Yeah.
Does it hurt the tadpole for its tail to be broken down?
I don't think so, but you never know. But I don't think so. All right, we talked a lot about mitochondria. And, every elementary kid knows it's the powerhouse of the cell.
It is.
But it's the powerhouse of the cell because what happens there? Cellular respiration. And we know it makes ATP, which provides the activation or free energy for pretty much everything you do, right? We know that the innermost part of this mitochondria is called the matrix. What happens inside that matrix? Krebs cycle, or what's the other name? Citric acid cycle. Step 3 of cell respiration happens on these little folds. These little folds are called cristae. And what's step 3 of aerobic respiration?
The electron transport chain.
Electron transport chain, also known as?
ETC.
No, the process is also known as? What kind of phosphorylation? Oxidative phosphorylation. Some unique things about the mitochondria. Remember, it has its own what? DNA. That's very similar in structure and shape to what? Bacterial DNA. Because where do scientists think we got them from?
We phagocytize them.
Bacteria that were phagocytized and taken in. What else do they have that they probably shouldn't? Got 2 membranes, which scientists think was the result of them being phagocytized and put in a vesicle around them. They also have their own ribosomes, which are much more like what? Bacterial ones than they are prokaryotic ones. And what can they do inside a cell that they probably ought not be able to do? Split. Sort of like what process? Binary fission. So all of those things that I just mentioned, what other organelle in plants have all those same characteristics? Chloroplast. And again, we think that eukaryotes got them by pulling them in. What's that idea? That a lot of organelles came about by pulling in other cells. Yeah, it's called the endosymbiotic hypothesis. And one of the main purposes of that endosymbiotic hypothesis is to show how that eukaryotic cells got to organelles, right? To show how eukaryotic cells came to be.
So mitochondria are in plant cells as well.
So you're saying that mitochondrial ribosomes are more like bacterial ones than prokaryotic ones?
Eukaryotic ones. Did I say it wrong?
Yeah.
Eukaryotic. Yes. Might have misspoke.
You said prokaryotic.
Gotcha. I misspoke. Make sure you can explain what the endosymbiotic hypothesis is. They like to ask about that on the DB exam. Also know some pieces of evidence.
Is that like the double membranes?
Yeah, that mitochondria and chloroplasts have double membranes. That mitochondria and chloroplasts have their own DNA. That they can do binary fission independently of the cell they're in. All those kinds of things.
So that's the kind of thing that the Baker response is on, like explaining it.
Yes.
Is that like the Baker response, or is it like?
It could be either one. I would say it probably more likely to be a big one, but it could be just a small piece of one. All right, we will end up today. Let's talk about this just very briefly. Extracellular matrix. What do we know extra means? Outside. So extracellular matrix is stuff that a cell makes and then does what with? Pushes out. and it ends up around it, the cell. Most of the time, what happens to it once it gets pushed out? It hardens. So this is like bone. You actually got cells that make the bone inside and then excrete the material and it hardens around it. Cartilage works the same way. What's collagen? It's a protein in your body that helps to hold you together. It's real strong. It's in like ligaments and tendons and even in your skin and the walls of your blood vessels. It's also an extracellular matrix. It's made by cells. They secrete it out around them, connect things up. Cell walls are actually extracellular matrix. In a plant, the cell makes cellulose, essentially spits it out around itself, and then it hardens into a wall. What are cell walls in things like fungi made out of? Chitin, different sugar. What about bacteria? What are bacterial cell walls made of? Yep, protein, peptido, and glycan, sugar. All examples of extracellular matrix. And I will end today by looking at this. Intercellular junctions. Inter means between. So these are ways different cells, adjacent cells, connect to each other. How do cells connect together? One kind of junction between cells are called anchoring junctions. And this is when the two cells essentially secrete this glue-like material that glues them together. The advantage of this is it's really stretchy. Maybe the two places in your body that you need really stretchy, well, maybe three places, where you need really stretchy connections. In your skin, when you think about your finger needing to be able to be in like a needs to stretch. Your bladder, pretty important that it can stretch. And in you ladies, your uterus starts out about the size of the fist. But let's say you have quadruplets one day. That thing is going to more than quadruple inside, and it's going to need to stretch.
Like it'll stay there.
It's going to stretch in all directions. That one uterus is just going to stretch
So like
out.
after you have kids or
Oh no, it'll go back. Probably not ever what it was, but it'll go back. Smaller. All right, so that's one kind of connection, an anchoring junction. Another kind of connection we want to talk about, and maybe the more important of these, are communicating junctions. There are certain kinds of tissues where the cells have to communicate with each other. We talked a lot about the heart. And the cells in your heart are called cardiac muscle cells, right? And if you took out a heart and you cut it into individual cells, and you kept those cells with nutrients and water, what would they continue to do? They continue to beat, but they wouldn't beat how?
at the same time.
They wouldn't beat at the same time. So that when they're together, when they're hooked together, the connections between the cells are called gap junctions. Because this is a type of communicating junction, gap junctions in animals. And that lets the cells communicate, and it allows them to get in rhythm so that they're all beating together. Yep.
How do the cells beat?
What do you mean? How do they beat?
Like, how do they like move like that? Like, what causes it?
It's essentially contractions, just back and forth contractions. And this gets pretty deep. But one kind of cool thing about cardiac muscle cells is they don't need what to beat?
They don't
ATP.
No, they need ATP. They don't need a nerve to tell them to beat. They're self-stimulated. Like my hand to do this, a nerve has to tell it to do this, right? But my heart, it does it
So
automatically.
if they go outside,
No nerves needed.
it can beat outside the body.
Can beat outside the body, literally. The whole Indiana Jones and the Temple of Doom, yeah, that can happen.
Wow.
If you could rip somebody's heart out, it would beat in your hand. Literally.
Can you give us a demonstration?
Come on up.
C30.
Anyway, that'd be good. The last thing I'll mention here, in plants, the communicating junctions found in a lot of plant cells are plasmodesmata. What communicates it, or what complicates it a little bit in plants? There's a wall. So this connection is almost like a tunnel through the wall into the cytoplasms of the two cells, and it'll allow them to transfer information, materials, one plant cell communicating with the one other cell. And that is a good place to press this out.
Some of my.
I have rough.